up, but I'm gonna put it a little bit later, and maybe in the meantime that Kim is gonna introduce herself, maybe we can just leave you a little bit of time to set up your R session. So welcome to our ladies event, and I'm really happy to see some old faces and some new faces as well. And to start, well, present. Okay, so this is what you will need to set up for the workshop. And I'm really happy that I would like to start, first of all, by saying a very, very big thank you to Zendesk, who keep hosting us for every event and gives us food and drinks and a beautiful space where we can keep holding our events. And it's, we are really, really grateful for their support. And as a start, I would like to mention that it would be great for who hasn't done it if you could please fill in our biannual survey. I know that maybe some of you are new, so maybe you haven't been to our events in the past. But if you have been at least to one event, you can still fill in this survey, which would really, really help us to improve our events in the futures. And you can find the link. I think we're going to put the link in Meetup, so you can find it there. But also we are going to put the link. Well, I put the link into our GitHub events into our GitHub page under the survey links repository, so you can find all the links of our surveys there once we update them or once we put new surveys. And so far, I think we opened this survey in July, and so far we only had 12 answers. But I mean, for sure, it is still good. But you might understand that it's not enough to have a overall idea of what we have done so far and what we can do in the future. And I must say that overall, these were very, very positive feedback. So in terms of the time of the events, in terms of the locations and the topics covered, we really had very interesting comments and positive feedbacks. However, there were also some comments and suggestions which are really important, because this is what helps us to improve in the futures. In the future. And in particular, we were suggested if we could, for example, provide an intro to our workshops, which is what we are going to do in September. And so I hope that we will meet your expectation with this. And also, we were also suggested to request to record events in case maybe some people miss the events and they cannot be present here. So by tonight, this is the first session that is recorded, so you will be able to find the record of the events, I suppose maybe on Meetup or on GitHub. We still have to decide that. And all the slides and materials are always available on GitHub at the same link that you saw before, which is also this one. So github.com R-Ladies AU events 2017. So all the events, all the past events and the new events and tonight's events will be there. It's also in one of the questions of the survey is if you maybe will need childcare for our next events. Maybe some of you do not need that, but it's also important for us to know, especially for the events that we would like to hold in September, which is on a whole day on Saturday, which maybe requires some women to leave their child somewhere. And it will be really, really good to know if we need to organize that, because it requires us to think a little bit more carefully of the space and people to call to do that. But we will be really, really happy to do that if you need that. We might think as well, once we organize the event for September, to ask you maybe 1 or 2 questions in terms of what are your needs, maybe in terms of diet requirements, or if you actually need a childcare at the event. Last thing, I posted this on Twitter, but I would like to remind you as well. And if you can share it, if you find this on Twitter and you want to share it, please retweet and share it on Facebook or wherever you want. So Stephanie Kowalczyk, which is one of our former speakers, I think she gave a talk in June about tennis, about using R and prediction models in sports. So she told us that they're opening 2 positions at data scientists at Tennis Australia, and she would really, really love if some of the R-Ladies would like to apply and try this position. So she will be really happy to answer any of your questions. Just send her an email, and if, for example, when you leave the event today, you don't remember who to email or what to do, just send us an email or a tweet or anything, and we'll be able to tell you all the info that you need. So the position is open until the 4th of September, so you still have some time. And okay, now I'm gonna leave the stage to Marie, who's gonna present the speaker for tonight.
So we are really happy to have today Dr. Kim-Anh Le Cao. So she has been doing her PhD in France, in the University of Toulouse, and then she moved for her postdoc in the University of Queensland. And she recently joined the University of Melbourne as a senior lecturer at the School of Mathematics and Statistics and the Center for System Genomics. So we are very happy that today she will tell us a bit more about her package that she has been developing during the last years, which is called MixOmics. And we hope that you all enjoyed this workshop. So please join me in welcoming Dr. Kim. We have some stickers, so if you are interested in the sticker, just comment and we will work.
Hi everyone, and it's a great pleasure for me to present some of the research work I've been doing that also includes developing an R package called MixOmics. So my talk is going to be, I think, in 2 parts. One is I'm going to give you a bit of context about the type of methods we have in MixOmics, how it works. And then either before or after the pizza, there's going to be 2 tutorials where we're going to run together some of the methods in MixOmics. And at the end, I'll give you some tips about if you're interested in developing an R package. So just a bit of background. So it's funny because in this room, there's 3 people who have been in the same school. I mean, what is the probability here? So it's an engineering school in Toulouse. It's a 5-year diploma. And from there, I then did a PhD, and then I moved to Brisbane. Then I started my group leader position in a biomedical institute, and I just recently moved to University of Melbourne. So just a bit of research context. Who doesn't work in biology here? So I just give you a bit of context to just so that you understand why we need to integrate this kind of data, but I'm pretty sure that the need is also in other fields. So in biology, we are able to measure different types of data on the same individual or the same patient. So for example, you can have different molecules, you can have information about cells, you can have information from the clinician as well, so clinical variables. And so that has really shifted a way of analyzing this kind of data, because before we had a very reductionist approach where we would say there's one gene that must be responsible for that disease, so let's test that gene, let's do that experiment for that gene. So we would have one hypothesis and we do one statistical test to assess whether that gene is responsible. Whereas with all these deluge of data, then we have to have a more holistic approach. So this time we try to understand all the different types of variables, of molecules that we measure globally to understand disease. And so this is why we need to integrate these different data sets. It's to understand really to understand better a biological system, and also to postulate new biological hypotheses in our case. And so to do that, there's different types of methods that can aggregate these types of variables. The one I'm specialized in are called linear multivariate methods. And so what they do is they aggregate these variables. into what we call components. And so I'm going to explain to you how that works. So here, variables can be genes, transcripts, proteins, but it can be anything you're measuring, can be clinical variables, any characteristic of the system that you're trying to describe. And so those methods are really useful because they reduce the dimension of the data. They can handle correlated variables, so variables that seem to explain the same thing, that usually breaks your statistical algorithms or models. And they also capture and explain variation in the data. So again, I'm going to explain what it means. And well-known multivariate methods are principal component analysis. They're very well-known in France. And also models called projection to latent structures, so PLS models. So I said that we were aggregating variables, and we do that by creating a linear combination of variables. So this is an example, a very small example here. I have 10 individuals, and I measure two variables. So for example, the height and the weight. Okay, so each individual here has two variables. So my data matrix is of size 10 times 2. And what you can do is you can decide to assign to your first variable, so for example, the height, you can decide to assign a weight, 0.2. And for the other variable, you decide to assign another weight. And then what you do is you apply those weights to each of those variables. Okay, so 0.2 times the first variable plus 0.8 times the second variable. So this is why we say it's a linear combination. It's linear because there's a plus. And it's a combination because at the end, you obtain one value for each individual. So one score, and that score summarizes all the variables you have measured. And so this linear combination is called a component in multivariate methods. And we need to decide what is the best weight to assign to each variable. Okay, so a weight like 0.8 is going to be more important, and it's going to put more weight for that variable because we think that in that model, that variable is important, whereas that one is probably not that important to describe an individual. And so we have methods to define those weights in an optimal way. And we also are able to work with large data sets with algorithms such as singular value decomposition. So that's the idea here, is that we go from a data set with two variables to a reduced data set of only one variable, which is your component. So I'll show you now an example that is a bit more complicated. So here we have 5 variables and we have 10 individuals. We do the same, so we calculate this linear combination. And if you are familiar with PCA, you will see that you obtain different components. Because so one component is a linear combination of all the variables here. And the second component is also a linear combination of the variables here, but it tries to explain something different from the data, just in case we missed it. Because you can imagine that here, I mean, this is we reduce the dimension here, but by reducing the dimension, we lose information. And so the components here are supposed to extract the remaining information from the data. So if you don't want to lose any information, you would have as many components as the number of variables that you have from the start. Okay, so five components for 5 variables. But the point is that you want to reduce the dimension of your data. And so instead, you're going to keep only the first few components of your model. Okay, and you say, this doesn't explain much in the data, I'm not interested at all, so I'm just going to have a look at these 2 components. And just remember that each value in your component is a score that summarizes all the variables that you've been measuring on a single individual here. And so usually, as I was saying, you keep or you retain a very small number of components compared to the dimension, the original dimension of your data. So here we have a dimension of size P, and P can be very large in biology, can be 1,000, 10,000, a million. And in the end, you obtain a very small reduced data set of, say, a dimension 2 or 3. And so PCA, so the example I was showing you before, is a matrix decomposition. So we compress the data. So we start with a data set of with a lot of variables and usually a small number of individuals or samples. And then we decompose it into a set of components and a set of vectors that are called loading vectors. And those vectors tell us how important those variables are in your linear combination. So for each loading vector, you have one component that is associated to it. And so when you calculate the PCA, all you need are these loading vectors, because once you have the weight, you can recalculate your components by calculating the linear combination. And in these loading vectors, and we know which are the variables that are important to define the components that reduce the dimension of your data. Okay, so we go from a very large data set to only a set of those vectors. So that's very easy then to work with those vectors. And so once you have those components, so each component describes one individual, and so you can plot one component versus another component, and that's going to represent your individuals or your samples in a very small space. So this is a space of dimension 2, and each dot here is your sample that was described by these thousands of variables. And so what you would like to see here so here we have a data set with 63 samples or 63 individuals, and they were described by more than 2,000 genes. And so in the end, we project the data into a space of 2 dimensions. And so here we have our individuals. And what we would like to see here is whether some individuals are more similar than others. So you would like to see some clusters of those individuals and see whether that corresponds to some disease or some type of tumor. So this is what we call an unsupervised analysis, because I never tell the method that I want to discriminate different tumor subtypes or anything that I measure on those samples, okay? So I'm just looking at the data, I'm reducing the data, and I'm asking what are the major sources of variation or sources of information that I can extract from those data. And so this plot tells me that those samples seem to be similar, those samples seem to be similar, and so on. But you also have another type of method where you include now the phenotype information or any information you have from your individuals. For example, if you want to compare sick people versus non-sick people, that's the information you would like to include in your analysis, because you want to try to explain why there are differences between those individuals. So here we have a big data matrix that describes those individuals, plus an outcome that tells us whether that individual is sick, very sick, or not sick, for example. And it's the same as the PCA I was showing you before. We decompose or we compress the information into sets of vectors. But the way we include the information about the outcome is that we maximize, or we try to correlate the data set we have with the outcome. And again, we do the dimension reduction. So this is one step of doing dimension reduction, is to compress the data. Another thing we do is we actually set to 0 some of the weights in the loading vectors. And so that means that the variables that are assigned a very small weight are discarded from our model because we think they're not important. And in biology, that's important because we can then highlight biomarkers or very key variables that seem to drive the differences between people who are sick and people who are not sick. So we can, having identified those biomarkers in these large data sets, then help us to do more experimental validation. And so this is how we perform what we call variable selection. So from a big data set, what is the small subset that is really important to explain the outcome? And then you can project your data into a smaller dimensional space like this. So it was similar to before, but this time I have included the tumor subtype information. So in that data set, we have 63 tumors, and we know that they belong to 4 different types of, subtypes of tumors. And I included that information in the PLS-DA model. And so you can see how the samples now cluster according to their tumor subtypes. And so because it's a sparse model, I can then identify which are the genes that actually explain the differences between the different subtypes of tumors. So in the tutorial that we're going to do later on, you will see the difference between a PCA that is unsupervised, so no information about the tumor type, and it just explains to you, what are the major genes that seem to drive the similarities between clusters. And you will see that it doesn't necessarily correspond to the tumor subtypes. But if you do include then the tumor subtypes information in your model, then you have a more directed analysis, and this is called a supervised analysis. So here we really aim to discriminate the different types of samples. So just to summarize here, those multivariate methods achieve matrix decomposition. So the PCA basically is going to try to maximize the variance of your components, so maximize the information we have from the data. Whereas the PLSDA is going to maximize the information we have from the data plus the outcome, okay, and try to relate this in some way. But there are other, and then we generalize it to more methods than that. So for example, you may have two data sets, for example, clinical variables and genes information on the same samples, and you want to see what are the clinical variables that are related to the genes, for example. In that case, we have another method called PLS that, again, decomposes the matrices into those components to try to extract the common information from those two data sets. So these methods, so we call it 2 omics because we have, in biology, we have different levels of different types of molecules that are called omics. And they can be integrated that way. So they were different. So the methods have been used by other researchers, whether it is in cell neck cancer, E. coli. This is the Terra Ocean data. It has researchers have used those methods to try to relate disparate or heterogeneous types of data. And from there, then we thought, well, where do we stop at 2 data sets? So let's go further. And so now, one of the latest method we have developed is to integrate more than 2 data sets plus the outcome. And again, you will see, so this method is called Diablo, and you will see that each data set is decomposed into sets of components. And we can then project the data into a smaller subspace. Okay, so this data set, this big data set, orange data set, for example, can be very large, and it's summarized into 3 dimensions, and the same for the green data set and so on. So you really reduce the dimension of the data. And on top of that, we perform variable selection. So we know exactly in these data sets which are the key drivers that drive the differences in our outcome. So I show you how flexible those methods can be. So for example, if you only want to analyze 1 data set, you can use those methods to explore, to do some data mining, dimension reduction, visualization. You can integrate multiple data sets. This is what I showed you before. So we try to find the common information from all these data sets. You can do unsupervised analysis where you just look at the data, you don't direct your analysis in any way, and you just want to see if you can identify a subset of correlated variables that seem to explain the same thing, even though you may not know what the thing is. And you also can do supervised analysis or supervised classification where you include in your model the information of your samples that the outcome you want to discriminate. And so this is really where my research lies, is trying to make sense of those big data sets by reducing the dimension of those data, but also by identifying the key variables in those large data sets so that we can reduce the dimension further. I'm going to skip this, and I'll show you this. This is a framework in MixOmics. So this is just one a sub-framework in MixOmics. So I'll show you there's actually much more methods than that. But we have a framework where we can analyze 1 data set, or we can integrate different types of data sets in different for different types of questions. But what really helps us is that all these methods are multivariate methods. So we actually rely on those components to then visualize the data. So the graphical outputs are very similar. Whichever method you use, you're going to always end up using the same type of methods. They are function S3 functions in R, so you always call the same name of the same function and you get similar types of outputs. And so you can represent the individuals or your samples, and then you can also represent the variables. So which are the variables that drive the differences between your patients groups, for example. The Diablo method that integrates multiple data sets is one of the framework. We have another framework called MINT. And the challenges with those data, and especially in biology, is that we usually have a very small sample size, a very small number of individuals, so usually 50, 100 max, and a very, very large number of variables, so more than 1,000 per data set here. And the problem is that those data sets measure different things. They're very heterogeneous data sets. You know, your clinical variables cannot be compared to gene expression directly. And so we have to deal with what we call unwanted systematic variations, a variation between platforms, between machines we use to do these measurements. In MINT, here it's a different problem because we try to integrate independent studies. So, for example, you may do an experiment in lab 1, an experiment in another country, and you want to combine those experiments somehow because you want to increase the number of samples or individuals you have. And again, you have this issue of unwanted systematic variation because one lab may use one protocol and another lab may use a slightly different protocol, even though they have the same question. So we call that framework P integration. Because here what we do is we try to integrate independent studies that are measured on the same P variables. For example, you may do a gene expression experiment and you want to combine those experiments on the same variables. Whereas the Diablo framework is called the N integration because here we measure different experiments that are performed on the same N individuals. Okay, so that's the difference between N and P. And the way we deal with this unwanted systematic variation is, as I told you, we project the data via the components. So we project the data into a smaller subspace, and we project them in a way that the different subspace actually agree. So it's as if it's a very abstract way of considering data. Is you project data into a completely different space. You may not even know where that space is, so that all your spaces of all your data sets correspond and agree. And by doing that, you remove the systematic variation from your data. So just a quick recap here. So linear multivariate methods, they look for components that summarize the information from your data. And each component is a linear combination of your original variables. We have developed methods to perform variable selection. We call those methods sparse methods. And just a technical term here, they use penalties in our model to penalize the weights that define our components. And the methods we develop are based on PLS, DA, and other sparse variants. Do you have any questions now? Okay. Yes?
Maybe when we will try with the workshop, we will be able to see. Is one of your able to understand, for example, what is the mechanism, like what is actually driving something, what is driving the group to cluster like that, or is merely to describe and to use the variables?
So you can do both. So if you want to just explore, describe, then you would do an unsupervised analysis. You just plug the data in, have a look at your samples, do they cluster, and why do they cluster? Yeah. So that's going to be describing only. But if you want to explain more and find predictors that are going, for example, if you want to develop a medical test that's going to tell you that person has cancer or not, Then you would do a supervised analysis, because that's going to really tell you what are the predictors, the variables that are going to predict whether that person is going to be sick or not sick.
Yeah, because well, if you integrate, so you you're not going to have every single variable at a time, but you're going to have things.
It's not together. So that's the trick with those methods. Is actually you use components to summarize for every data set, knowing that those components from one data set are correlated or are related to the components from the other data sets. So that's why we integrate. So it's not just about combining, it's trying to find the key variables in those data sets that seem to explain the same thing, even though they completely, yes. So the idea is that then we would understand a bit better the biological mechanisms at different levels, molecular levels that we measure. Yes. So you will see, so hopefully we have time to go through this, but in the Diablo example, you will see, we have mRNA, microRNA, proteins, and you will see we have this signature that seem to explain an outcome. Okay, we're going to go for pizza, but I'm just going to show you. It's really important to have the Pizza Hut here. So there's going to be two examples here, and this is just the key figures that you will obtain. And you have all the RMD and the R codes available on GitHub or on my slides. And we're going to run it and just discuss some of the results.
You were very quick. I think I did a very good introduction. Everyone has so time.
Yeah, I mean, so we have so if we do 20 minutes,
yeah.
That leaves us about an hour.
Yeah. I mean, yeah, I think to be more, because I mean, we have 20 hours here.
Yes, when do people
So we can be there till 7. I think normally it's
so I think if we finish the track at 7.
Yeah. Maybe it's good. So if people want to stay here and want to ask some more questions or talk a bit about it, it would be good. No, but yeah, it's really good. Thank you. Yeah, I haven't tried it yet because I haven't had a chance to apply with my data, but yeah, I'm really good. I remember, I've seen already 2 talks from you, but I've never actually tried it. And I'm like, okay, now I'm going to try it. So that would be great.